right here. Real life is people sometimes come in during your training, right?
Mm-hmm.
All right. So let's
Say what?
is everybody ready?
Yep.
All right.
Go.
Go.
Okay. Hi, my name is Jill Moore. I'm an applications support specialist, primarily focusing on the Kingfisher instrumentation and kits. I've been with Thermo since about 2007, supporting or selling. I did sell these in the field for a short while, the Kingfishers, most of that time. So what I'm going to talk to you about today is how this technology works. This is a kind of an overview of the whole product line, but mainly how the technology works, because you're going to have to know that to sell this to your customers. And then we'll do separate presentations for each model that will have more specifics. But all of these instruments work on the same technology. It's just a matter of how many samples they process and how they differ. So what are Thermo Scientific Kingfishers? They are magnetic particle processors that purify macromolecules such as DNA, RNA, or proteins, or cells, whole cells, from some starting material that a life scientist wants to work with. Why would a customer choose an instrument to do this? Because you probably know that there are ways to do this manually that are very, very common, column-based methods, even magnetic particle methods that are done manually. Why would somebody want to do this? Has anybody worked with automation before? Anybody? Okay, what does automation generally do for your customer?
Frees up time to do other things.
Bingo. What else?
Reproducibility.
Reproducibility and consistency in results. Exactly. So there's two payoffs for the customer to automating anything with any kind of robot. It takes their hands off the process. That gives them walk-away time to do something else, so you get more bang for your buck when you hire a tech. And it makes the results more consistent and reliable because you don't have that human error factor. There's no differences in the way I pipette from the way you pipette. Okay. So it all starts with a magnetic bead, B-E-A-D. Each of these particles, which are commonly referred to as beads, contains an iron core. This is what makes them magnetic, of course. The iron core is surrounded by a polymer capsule so that the metal doesn't touch your molecule. You don't want the metal interfering with the charge on your molecules. And then around that capsule, you have some kind of coating that attracts your target molecule that you want to purify, okay? The optimal size to work with the magnets that are on our instruments is about 1 to 10 microns. That's important. There are smaller beads on the market. They do not work with our instruments, but fortunately for you, probably 90 or 95% of the reagents on the market do have this bead size. The coating around the outside, as I mentioned, is specific for whatever you're purifying. For DNA and RNA, that's typically silica because it's easy to make those molecules bond to silica under high salt conditions. It's the exact same thing used in a column. It's just on a bead. For proteins, it's kind of all over the map. You would coat it with something that attracts the protein you're trying to purify. So it might be something that attracts a tag that you've cloned into that protein. It could be an antibody specific for that protein or an antibody specific for another antibody that is specific for the protein. So there's all kinds of ways to do this. We also sell blank beads. So just like you would coat an ELISA plate to pull down specific proteins, you can coat a blank bead. And Dynal is the most popular brand. It's now part of Thermo. So how does this happen? Basically, there are 3 steps in a magnetic bead purification. Whether you're doing this in a manual process with a benchtop magnet and a handheld pipetter, or whether you've automated it on a Kingfisher or on some other kind of liquid handling robot, which is also possible. You bind your target under specific buffer conditions. You wash it, maintaining those same type of buffer conditions to get rid of any contaminants. And then at the end, you put it in a buffer that will allow the bond between your target molecule and the bead to break. So that then you remove the empty beads and you're left with purified, for example, DNA. How does the whole workflow look? You take your starting material, something that contains DNA or a protein or a cell, and you mix it with your magnetic beads in specific buffer conditions. Binding takes place during an incubation and mixing period. Once it's bound, 2 to 4 washes is typical for nucleic acids. At the end, you put it in your elution buffer that breaks the bond, and then remove your empty beads. So to perform an extraction of, say, 24 samples on a column using individual columns, which is how a lot of people do this, takes about 2 hours. On a Kingfisher instrument, to get DNA takes 30 minutes. You can do those same 24 samples in 30 minutes versus spending 2 hours. And most of that time is on the instrument. You're not even doing anything versus the very hands-on column process. Magnetic bead extraction came about in the 90s as a more straightforward, less time-consuming manner than columns. So you can do this manually. If you were to automate this on a large robotic liquid handler, where's my doohickey? There it went. It would basically look the same as this. So the first step, you mix your sample with your beads in a well or a tube, bind it, mix it up and down, or the liquid handler would be doing the same thing, the robot. Then you would pull the beads down to the side and change the buffer. So every time you change this liquid, you leave the beads stuck to the magnet, and then you take it away from the magnet, put fresh buffer on, wash it again, so that you're drawing the beads down every time and changing the liquid. On a Kingfisher, we don't change the liquids. We move the beads. So once you bind your target to the beads, you use magnets, magnetic rods on the instrument, to move them from one step to the next. And the next thing we're going to do is hopefully this will actually work. Where's my doohickey? There you are. So we're just going to see a video that kind of illustrates how this happens. That's some nice little music. I don't know if you guys can hear it. It kind of puts me to sleep. So here you see a magnet pulling beads out of solution and moving the beads into the well where the sample resides. The DNA binds to the particles. Typically, the magnet's going to be moving a lot faster than this, but just for illustrative purposes, the animation goes a little slower so you can see what's happening. The DNA is then washed. You can leave the beads stuck on the magnet if you want, or you can have the magnet pull out of the sample to have it mixed more efficiently. This takes place 2 to 4 times depending on what you're purifying and what your starting material is and what reagents you're using. It will vary a little bit. Then at the end, you put your beads into the elution buffer. The bond between the beads and the DNA is broken. Then the magnet comes in and removes the empty beads, leaving you with purified, concentrated target molecule. So we're going to skip out of that. And go back to the presentation. So just to summarize that process, kingfishers move beads instead of liquids. This gives you superior purification and high quality concentrated end product, because over here you're either leaving behind some liquids so as not to sacrifice your yield, or you're leaving behind some beads to get greater purity. So here we have data showing how high our particle movement is. This leads directly to increased yield. Why would you think moving the beads is important?
Okay, so this is where you would have a quiz and have people
Question.
answer the question?
Just have to
Okay.
answer the question. This slide, where you can find opportunities everywhere. What kinds of materials are these scientists working with? Just about anything a life science would want to work with, you can find an opportunity in that lab. We have 4 models for different levels of sample throughput with reagents and kits.
Let's, I don't know if you have
Yeah.
is this your summary slide? Do you have a summary?
Yeah, well, sort of why were the instruments named Kingfisher? Because they have beaks that go down like the magnets. So, yeah. So, thank you for your attention.